So, hello everyone, and these are the second lecture of our discussion. Unit 13, methods in biology. These are actually deals with the techniques, molecular techniques, and we'll discuss all the previous year question papers of CSR net syllabus. And you have to be careful on that. The CSR actually asks many twisting questions, experimental based question. In methods in biology, you must have to know the basics of the all the methods, what's the purpose of the method, how it is used for a research purpose, right? So we have discussed a few questions in previous lecture. Now in the these are the second lecture. We'll discuss the from we have discussed already this one. If you can back this lecture also, uh, I'll give it in the description below, so you can able to watch this discussion. Now this from that the question number 3, this will be the starting one. Now, fluorescence recovery after photo bleaching in live cells is used to determine. Now, in case of FRAP is stands for fluorescence recovery after photo bleaching. Now, in case of FRAP experiment, we used to actually the when during FACS experiment, we bleach the particular area by laser beam. Laser beam is applied and bleach the area, and we observe the rate of recovery of the bleached area, how fast the bleached area is recovered. Based on that, we calculate the fusion constant, right? And how much the proteins are mobile, or diffusion of the rate of the protein can be calculated, right, on the basis of FAC experiment or fluorescence recovery after photo bleaching. So from that, the fluorescence recovery after photo bleaching, from bleach area, we will plot a particular graph, and from graph actually represent x-axis time and y-axis the fluorescent intensity, fluorescence, fluorescent intensity. From that, you will able to from graph you will able to analyze the data, that how much fast the is recovery is occurring. Suppose these are the rate of recovery and recovery based on the either the proteins is mobile, or it's a freely diffusible protein, then its recovery rate will be faster. If it is a protein is integral protein, integral membrane protein, then the recovery rate will be slower, right? So here from that, fluorescence recovery after photo bleaching in living cells used to determine co-localization of the protein. No, actually it's not co-localization, distance between two organelle. It measures the diffusions of the protein, right? Fluorescence recovery after photo bleaching measures the diffusions of the protein, and from that the different graphical analysis from that graph also came in previous question. Suppose you have if they will give the different 3 or 4 graphs, and from that you have to predict which one is the integral protein, which one is a freely diffusible or diffusion rate is higher, and from that you can calculate the diffusion rate of the protein, right? And either the protein is membrane bound or freely diffusible can also be predictable by the FRAP experiment. Now, second, this question, actually you have transiently expressed a new protein for which no antibody is available, right? In a cell line to establish structure function relationship, which one of the following strategies is the most straightforward the way to examine the expression profile of this new protein? Now, see, you are doing an experiment and you have transiently expressed a new protein for which no antibody is available, right? And in a cell line, now for that, you know that during western blot or sds page or sodium dodecyl sulfate polyacrylamide jelly electrophoresis, actually uh, the separation of the protein based on their molecular weight, and the sds actually make the protein uniformly charged so that the protein will migrate in a particular one direction only, right? So that and for that SDS page you must have to use antibody, right? So running SDS page identifying the protein, you need antibody, but the question is said for which no antibody is available, right? So antibody is not available for that protein. Now you have to do that experiment and you have to determine the expression profile of the new protein. Now by metabolic leveling of the S35 labeling amino acid, in case of in immunoprecipitating this protein with help of another protein for which antibody is available. Now for immunoprecipitation you also need antibody, right? Immunoprecipitating this protein with the help of another protein for which antibody is available. So this also not be correct because the antibody is not available. So another is that in metabolic leveling with S35 labeling amino acid. Now S35 labeling amino acid, but you have to, you should know that the protein is what's the sequence of the protein, but you don't know in question in the this is a new protein for which you don't know the sequence, right? And the straightforward, most straightforward way to examine is the making a GFP fusion protein with this new protein, because if you give the GFP as a, you know that glycan fluorescent protein or GFP during different experiment we use GFP as a reporter gene, and GFP expression, from GFP expression we, if it's tagged with new protein, suppose this is a GFP tagged and these are the new protein, if the new protein, now GFP, if the GFP is expressed, because then you will know that your new protein also is present there, right? Expression protein also expressed there, right? That from that way you can analyze that at the new protein, right? Because the question is asking there is no antibodies available and these are the new protein and you don't know the sequence of the protein, so how you level the metabolic using by S35 level amino acid, and it's not a straightforward way, right? The most straightforward way is the making a GFP fusion protein with this new protein, and GFP acts as a reporter gene. Now next question we'll see. Now, during an experiment, a student found increased activity of a protein for which there were three possible explanations. Increased expression of this protein, okay, increase expression of the protein, increase phosphorylation, or increase interactions with other effector protein. After conducting several experiments, the student concluded that increased activity was due to increased phosphorylation. Which one of the following experiment will not support, provide the correct explanation drawn by the student? Now you are a researcher and when you are doing an experiment and a student found activity of a protein, right? And suppose we know that we all know that DNA to produce mRNA and mRNA produce protein, right? Now this protein, and you have to analyze that protein, and then this have a somehow, that's this protein have increased expression level. That means in protein level there's a increased expression of the protein, increase phosphorylation. Maybe there is a post-translational modifications occurs in the protein, and for that reason, the post-translation there is a phosphorylation occurs, and for post-translation modification, the increased activity is detected, right? And now, the first of all, not support, which not support or provide the correct explanation drawn by the student. Now, where first one is Western blot analysis, analysis of transcription rate, mass spectrometry, and phosphoamino acid analysis. Now, if it is a post-translational modification or PTM or post-translational modification can be detected by the mass spectrometry. Mass spectrometry we use for post-translational modifications like phosphorylation, methylation, sulfonation, and can be analyzed by the for protein, and protein post-translation modification can be analyzed by the mass spectrometry technique. So mass spectrometry can be used. Now phosphoamino acid analysis, that can be used because phosphate here, phosphate actually added, so phosphoamino acid analysis can be used. You can do Western blot also if it's a protein is a and increase activity and the protein is phosphorylated, you can analyze by the Western blot also. But in case of transcription rate, if you analyze the transcription rate, you will not be able to analyze the protein expression, because transcription is you are doing the experiment in mRNA level. So if you do experiment in mRNA level, you will not be able to predict whether the post translation modification occurs or not. You cannot be detected by this and post analysis of the transcription rate. So answer is option B is the uh, possible answer. Option 2 is the correct answer. So we'll see the next question now. First one, a researcher is studying the subcellular localization of a particular X in an animal cells. The researcher performs several centrifugation in increasing rotor speed. The researcher starts spinning the cellular homogenate at 600 G for 10 minutes. Collect the pellet, spin the supernatant at 10,000 G for 20 minutes. Collect the pellet and spin the supernatant at 1 lakh G for 1 hour. Collect both the pellet and the final supernatant, one subjecting various plate and the final supernatant to Western blotting with anti-protein X antibody. The protein X is observed to maximally express in pellet after centrifugation at 10,000 G. Now, based on the above observation, what will be most likely localization of the protein X? Now, so these are big questions. So these are, you can easily observe the supernatant and pellet, right? During centrifugation we'll get a supernatant and pellet. So now you can see that the here that first is cell homogeneity is there 600 G. If you centrifuge, then you will get nuclei and cell debris, right? And from that, if you put 20,000 G or greater than, here a question is asking for 10,000, and you can put here 10,000 G, also 10,000 G. And if you put 20,000, you will get mitochondria, peroxisome and lysosome. If you put 1 lakh G, then you will get microsomes, and then from that you will get, next is cytosol you will get. So question is asking, after western blotting on subjecting various plate, final supernatant to western blotting with anti-protein X antibody, right? And X is observed to be maximally expressed in plate after 10,000 centrifugation. Now here 10,000 centrifugation, you will get mitochondria, right? Nucleus will get only, because nucleus is heavy, so nucleus will come first. So only 600 G is necessary for nucleus, so nucleus is not possible here. Ribosome will get it after 1 lakh G, so ribosome is not possible. Mitochondria and microsome also 1 lakh G, so mitochondria is the answer, right? If you remember this chart, you will be able to solve that question. So if you have a cell homogenate and if you put in centrifugation tube and 600 G, then you will first get nuclei and cell debris. In 20,000 G, for 10,000 G you will get mitochondria, 20,000 G you will get peroxisome, lysosome, and 1 lakh G you will get microsome, right? In case of ribosomes, all others you will get in the 1 lakh G, after 1 lakh G centrifugation, right? So next question we'll see. Match the following technique with appropriate application. Use from the list of the option given below. Now here is a CHIP. CHIP is actually chromatin immunoprecipitation. CHIP stands for chromatin immunoprecipitation. Chromatin immunoprecipitation, or CHIP, actually used for chromatin immune precipitation. And the bisulfite sequencing, we know that bisulfite sequencing actually used for analysis of the methylation sites, right? You can I have made a lecture or video on that, that bisulfite sequencing for DNA methylation analysis, and how we can analyze the methylation status of our genome, whether the CpG island in our genome are either methylated or not, how we'll predict, because methylation have connected with many diseases. There is a connection with the methylation of the genome, because most of the cases methylation in the CpG island that leads to gene suppression, or it's reduce the gene expression level. And that's actually bisulfite. You can watch that one also, bisulfite sequencing for analysis of methylation sites. First option, CHIP. CHIP stands for chromatin immunoprecipitation. CHIP is stand for chromatin immunoprecipitation. Chromatin immunoprecipitation is a powerful and versatile technique used for chromatin immunoprecipitation used for probing protein DNA interaction, protein DNA interaction within the natural chromatin context, within the natural chromatin context of the cell. So now you can see that chip is here. So chip will be identifying sites, so binding site of transcription factor, right? So transcription factor actually you can be analyzed by chip, chromatin immunoprecipitation. Now ELISA stands for enzyme linked immunosorbent assay. What is ELISA? ELISA stands for enzyme linked immunosorbent assay, right? Here we will use the actually most of the proteins detection will done by ELISA expression level, right? Protein expression level by using the antibody, primary antibody and secondary antibody. Then we use this technique actually used for detection of the protein expression level, right? And in res RLM res, the actually it's a use for removing the 5' transcription site, the full length RNA strand are treated by TAP. TAP stands for tobacco acid pyrophosphatase. TAP to remove the 5' cap structure and pyrophosphate, 5' cap structure and pyrophosphate and a RNA adapter and then a RNA adapter is directly ligated to the RNA 5' end using T4 RNA ligase, right? Now here, so RNA race actually used for identification of 5' and 3' end of the transcript, right? ELISA here is the quantification of transgene expression. So answer is option A is the correct match, right? Now next one. Now next one we'll see that matching the following. Now it's a functional magnetic resonance imaging or fMRI, functional magnetic resonance imaging, positron emission tomographic scanning, computed tomography and electroencephalogram, right? EEG, electroencephalogram, actually used for the different brain wave activity or electrical activity of the brain wave. If we can from that is EEG, we can analyze the alpha, beta, gamma, delta waves in our brain. Different types of brain wave can be analyzed, or electrical activity of the brain wave can be analyzed by EEG. And fMRI actually the functional, it's different from magnetic resonance imaging, is that here the actually we can detect the which area of the particular brain and actually more active during a particular task can be analyzed by the fMRI or functional MRI. During functional MRI detects the changes, the blood oxygenation on a, and flow due to neural activity, right? Based on and this detect changes in the blood oxygenation and flow due to neural activity. Now coming to electroencephalogram. Electroencephalogram measures the voltage or electrical activity of the brain, right? Now computed tomography or CT scan actually used for CT scan actually used for show imaging of the brain by differential absorption of X-rays, right? Different image of the brain obtained by the differential absorption of X-rays. And positron emission tomography use short-lived radioactive elements material to map the functional process in brain, right? So that's why PET is less because here radio short-lived radioactive materials actually used. That's why it's a less it's a more toxic than fMRI or functional MRI, right? uh, so answer is option B is the correct match. Now, now here a student and was asked to design a knockout cassette for a student was asked to design a knockout cassette for specifically deleting the P53 gene, specifically deleting the P53 gene for the prostate gland of mice. Which one of the following pairs of cassette will ensure the deletion of the gene? Now, suppose you are performing an experiment and you are working with prostate cancer. Suppose in prostate cancer and there is a mutation in the suppose in the somehow the you have to delete the P53 gene from prostate gland of mice. Now you have to design the experiment how it will delete the gene and now we know that during gene knockout experiment and LOXP if the direction in case of a pre-LOXP deletion system, if the LOXP in the same direction, if you design the construct in such a way, LOXP in the same direction, LOXP and LOXP in the same direction, it will leads to the deletion of the gene. Suppose gene is here, gene X, or here gene is P53. It's a tumor suppressor gene. So if you have to delete the P53 on the genome, and you can do that by placing the LOXP site in the same direction, right? So it leads to deletion. And if it is a LOXP in opposite direction, suppose LOXP in this direction, another LOXP this direction. So if the direction of LOXP, another direction of the LOXP is the opposite direction and gene is here, so it leads to the inversion of the gene, inversion of the gene, right? And in case of if the LOXP in two different segments of the gene, suppose LOXP, one LOXP is here, another LOXP is here, another segment of the DNA. So in that case, LOXP, that's LOXP, it leads to translocation. It leads to translocation of the gene. Now here, which one of the following pair of cassette will ensure deletion of the gene? Now you have to delete the gene. Now here you have to need that CRE TSP. CRE stands for CRE recombinase and TSP stands for tumor specific promoter, tissue specific promoter, and these are LOXP site, right? Now first option, look at the first option. Look at the first option. These are direction, same direction, This is a p53 gene you want to delete. Now these are Cre recombinase. Cre recombinase is actually necessary, and Cre recombinase is placed in tissue specific promoter, right? And these are the perfect construct, because if you have a tissue specific promoter is there, tissue specific promoter actually able to synthesize the Cre gene, because Cre have to also synthesize, right? It's need promoter. We all know that during gene expression, this must have it's a particular gene if you have to express in the protein level or functional level, because Cre is a recombinase, it is an enzyme, it is a protein, and it must have to synthesize from a particular Cre gene, and this Cre gene must have to transcribe, then make mRNA, then mRNA produce protein, right? That's TSP, that is tissue specific promoter, must be there for expression of the Cre gene, right? So Cre gene must be in the nearby of the tissue specific promoter, because a promoter region where RNA polymerase will bind and synthesize the gene transcription rate. Now here, look at the option 2. Now Cre recombinase is there, p53 is there. So if you have to delete the p53, p53 should not be here, and there must be tissue specific promoter. So that's why option 2 is not correct. See that tissue specific promoter Cre is there, but here loxp, direction of the loxp. Look at the direction of the loxp. Loxp direction is opposite direction, right? These are this condition. One lox p is one direction, another lox p is another direction, so that's actually leads to inversion. Instead of you have to delete the p53 gene, so that's why this option also not correct. Now you can see that option 4, tissue specific promoter Cre is there, okay, but the lox p outside of the target gene. If you must have to put your target gene in between the lox p site. And if you put the loxy site in other than your target gene, the gene will not be deleted, so there will be no knockout, so the gene will show. This also not correct. So that is the uh, for loxy for gene deletion on gene knockout experiment, and you must have to put the target gene and in between the loxy site, right? And loxy should be in same direction. If loxp is same direction, it leads to deletion. Loxp in opposite direction leads to inversion of the gene. Loxp in a different location, it leads to translocation of the particular gene. And now here gene should be placed in between, right? And it must have a it's Cre recombinase also necessary. Cre recombinase Cre recombinase also should be placed in between the tissue specific promoter. Uh, that's why option 1 is the correct. Now next question we'll see. Now, in unfortunately, the two proteins were found to co-migrate in SDS page profile. Under this situation, using one-dimensional SDS page by Western blot, which one of the following best way to demonstrate the presence of both protein, right? Now, suppose you are working with a particular protein, particular in a tissue, in particular two different proteins, right? And you are running the SDS page, and your observation is that the two proteins are co-migrated. They are coming in same in the jail run. During jail run, the two proteins are coming simultaneously in the same region. Now there both proteins are coming in the same region. Here, suppose here is coming the after migration, it will coming in the same area, both protein. Now by one-dimensional SDS page and by Western blot, which of the following the best way demonstrate the presence of the protein? Achha, developed Western blot with their antibodies in the same gel. Now it's as the protein is co-migrated and in the same area, right? And the prior to doing SDS page and Western blot, one protein could be removed by immunoprecipitating in the cell extract and silencing the expression of one protein at the time or by siRNA and performing Western blotting, subjecting the technique of striping and reprobing of the gel after transferring to the nitrocellulose membrane while doing Western blot. Now, this is the first one. Now, develop Western blot their antibodies on the same gel. Now, as they are co-migrating in the same area, and if you're using the antibody for the and for that protein, and they will not be able to, you will not be able to detect, because the two protein are in the same area, same co-migrated, so that's why they are not be equally bind with that antibodies, right? And prior, so that's why this is not correct. Prior to doing SDS phase and western blot, one protein could be removed by immunoprecipitating in the cell extract. Now, during you were running western blot and you trying to immunoprecipitate by using, by removing immunoprecipitate by remove the immunoprecipitating in the cell extract, right? Now these are the best way, because you can do that, but it will take time consuming and more costly, right? Silencing the expression of one protein at the time by siRNA and perform western blot. Now silencing the protein at time of siRNA, now because you are running SDS page, so these are SDS page, so how here we will not able to do that, right? If you have to silence the particular gene expression, and then you have to go into the mRNA level, and siRNA, miRNA, or those are microRNA actually makes the double stranded RNA and making the gene silence, so that cannot be possible here. Now you can see that striping and reprobing. What do you mean by striping and reprobing? Striping and reprobing is a western blot is a method in which primary and secondary antibodies are removed from a western blot so that the blot can be reprobed, right? A blot may be striped or reprobed several times to visualize other protein or to optimize detection of the protein, that is antibody concentration, without need for multiple jails and transfer, right? From that you can do that at a for that the answer is option 4, subjecting the technique of striping and reprobing of the gel after transferring to nitrocellulose membrane while doing western blot. Now, next question we'll see. Now, in case of several fusion construct were developed to purify heterogeneous protein in E. coli, that the table below is the fusion partner and the ligand. Now, here you can see that maltose binding protein, streptavidin, glutathione S transferase, flag tag, and 6 histidine tag. Now, in case of, we know that in case of 6 histidine tag, 6 streptavidin actually work bind with avidin. Streptavidin and based these are the streptavidin will be avidin. And avidin or biotin, right? Here avidin is contained, so streptavidin is biotin, right? Now, now is the his tag. Histidine 6 histidine tag is for nickel. 6 histidine tag actually bind with nickel. 6 histidine tag actually bind with nickel, and so another is the maltose binding protein. Maltose is a carbohydrate, so it's bind with amylase. So first is the 1D, streptavidin bind with biotin, 2E, glutathione S transferase. Glutathione S transferase bind with glutathione, and flag tag actually for a flag tag specific monoclonal antibody, and histidine tag for nickel, right? So option 3 is the correct answer. For in case of histidine tag actually bind for nickel, flag tag for specific monoclonal antibody, streptavidin for biotin, and maltose binding protein amylase, right? So hopes makes this clear. Thank you.